So today, we are going to discuss about the structures of DNA and DNA replication. DNA, or deoxyribonucleic acid, is a complex molecule that contains all of the information to build and maintain an organism, and it is used to produce and pass along a genetic material to their offspring. Our body contains cells, and every cell has a nucleus where the DNA is located. The structure of DNA. In the cell, we can find the double-stranded DNA, or the two strands intertwined to one another to form double helix, or commonly known as B-form DNA. Each strand in the DNA is called polynucleotide, meaning the polynucleotide is made up of individual units called nucleotides. The nucleotide has three components: the 5-carbon sugar, phosphate group, and the 4 nitrogenous bases, which is the adenine, guanine, thymine, and cytosine. Sugar is called deoxyribose because it is missing a hydroxyl group which are present in ribose. That's why the nucleotides in DNA is called deoxynucleotide. Nucleotides attach to one another in the DNA strand called phosphodiester bonds. Thus, the sugar and phosphate groups are the backbone of the DNA. Nucleotides come together through covalent bonds in the backbone. The two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base in the opposite strand, and each unit is called a base pair. The geometry of the base pairs in both strands is the same, allowing for symmetry and base stacking in the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures for about 10 base pairs. The regularity of helical structure forms two repeating and alternating spaces called minor and major grooves. Grooves acts as a base pair's recognition and binding sites for proteins. Major grooves contains base pairs specific information. Minor grooves is largely pairs non-specific. Because of the patterns and bond acceptors and donors interact in the grooves, the DNA can be acted upon in either or non-sequence specific manner, allowing protein to position themselves correctly in the genome to carry out their designated tasks. The DNA replication. DNA replication is a process in which the DNA is making a copy of themselves during cell division or making more DNA. DNA replication is important because it repairs old damaged tissue, and it will ensure that the new cell receives its own original copy of the DNA. DNA replication happens in both types of cells, which are the eukaryotic and prokaryotic cells. In eukaryotic cells, it happens in the nucleus. In eukaryotic cells, it's going to happen before the mitosis or meiosis in the time known as interphase. An interphase prepares a cell for mitosis by producing proteins and duplicating chromosomes, the key players in DNA replication. There are 4 kinds of key players in cell division, and most of them are enzymes that has the ability to speed up and break down the items involved in cell division. First is the helicase. Second is the DNA polymerase. Third, primase. And the fourth, ligase. The helicase. Helicase is called the unzipping enzyme. It unzips the 2 strands of DNA. It breaks through hydrogen bonds that holds the DNA bases together. The DNA polymerase. The DNA polymerase. The DNA polymerase is called the builder enzyme because it replicates DNA molecules to build a new strand of DNA. The primase. The primase is an enzyme that is called the initializer since it will help or make the polymerase's path. Next is the ligase. The ligase will help in gluing the DNA together. The process. DNA replication starts at a certain part of the cell called the origin. A certain DNA sequences is at the origin. First, the helicase comes to unzip and unwinds the DNA strand, then the SSB or the single-stranded protein binds to the DNA to keep the 2 strands separated. And then the primase will enter next to make the RNA primers to both strands to make a path for the DNA polymerase. Then the polymerase will build the new strands of DNA which the primer made. And since the polymerase can only build a straight strand, which is the 5' to 3' direction, that is called the leading strand. The direction 3' to 5' direction is called the lagging strand. It will create a fragment called the Okazaki. To keep off the fragments, the ligase will now enter and takes care of the gap by gluing or sealing them together. And then, after this process and replicating, you will now have a 2-identified double helix DNA. The end. Thank you for listening.